Hi, my name is Holly Woodward, and I'm a paleontologist. I have always been interested in dinosaurs specifically my whole life, as long as I can remember. And so that's what I wanted to do. That's the career I pursued through school. I learned as much as I possibly could about prehistory, so not just dinosaurs, but anything involving ancient life, and geology. And when I went to college, I focused on geology and paleontology, studied biology, and with my master's and PhD degrees, I've been able to do a lot of field work. So that's fulfilled a dream of mine, is to actually get out and excavate fossils. And of course, my first love was dinosaurs. So I've been on many dinosaur fossil excavations and currently lead one pretty annually in Montana. But that has been sort of an ongoing or continuation of my PhD research, which was on Maiasaura, which is a duck-billed dinosaur from the Campanian to Medicine Formation of Montana. And, so I've been really interested in studying how that dinosaur lived and grew up. And that has sort of led me to study other dinosaurs in a similar way, trying to understand their growth history. And this has led to more recently some studies on Tyrannosaurus rex growth, from looking at how they were growing as juveniles to achieving their really large adult size. But I've been other places to study other animals, and this is because I'm a paleohistologist specifically. So I do need dinosaur and other extinct animal fossils to do this, which has allowed me to, again, fulfill some dreams of mine and traveling to different continents, including Asia and more specifically Mongolia, to dig up Psittacosaurus specimens, which are tiny plant-eating dinosaurs. More recently, I was able to travel to Egypt in Africa, and apart from doing the typical touristy things, I was mainly there to assist and study whale and other marine mammal fossils. So just because my first love has been dinosaurs, with paleontology you really can do a variety of things. It opens up opportunities for you to go to many places, and you start collaborating with people that you may not have actually ever thought about collaborating with before, or you didn't expect to collaborate with, on projects to address specific questions that are appropriate or actually span not just one species or one group of animals, but questions that can be addressed to answer a whole bunch of questions from deep time. And specifically, what's really cool about the Egyptian research that I'm embarking on is that the Sahara Desert used to be underwater. It used to be an area of shallow sea, and so you actually do find whale fossils in the Sahara, which is really cool because that really ties into climate change in a way that people can see. So what does this have to do with polar dinosaurs? Right now I've been talking about deserts and dry places. Well, since I'm a paleohistologist, I take a look at fossil bone, and I actually look through the fossil bone, and I look into the bone tissue microstructure with a microscope. So it's really funny for me because I will go to these places, these dry, arid places that used to be completely different environments, and I will collect fossils from the animals that used to live in these different environments. And some of them, like T-Rex, has very large hollow bones, and you use a microscope to look at the teeny tiny little structures in this giant animal's bones. And so that's what I am interested in doing, and understanding how animals lived. So as far as polar dinosaurs go, bone histology is a way to look within the bone at the bone tissue structures, because as you grow, your bones change, and your bones record basically the life history of you or other animals. And so from looking at this bone tissue, we can figure out from extinct animals how fast they were growing, whether or not they were adults, how old they were when they died. And so specifically with polar dinosaurs, we know dinosaurs are found on every continent. So what made them so successful? And especially the ones that were living near the poles, both the North Pole and the South Pole. Temperatures weren't as extreme back in the Mesozoic as they are today at the poles, but they were still pretty unique. And so how are the dinosaurs at the poles adapted to living in these places? And so histology can tell us a lot about the growth and physiology of these animals that were living in these unique places. And more specifically with the polar dinosaurs, I studied a sample set of dinosaur fossils from Australia. Now, Australia used to be connected to Antarctica, and the two started drifting apart, but they were very close to the South Pole. And because Antarctica is still hard to get to, it's, you have to prepare for such a field season. It's very expensive. The fossils are in really hard rock. People are going to Antarctica and they're learning lots of cool stuff, but Australia fossils are a little bit easier to get to and can be a good starting place to look for fossils that may be from similar animals to those within Antarctica, and allow you to start asking some questions and building up data so that you can embark on these more rigorous field expeditions to places more difficult to reach, like Antarctica. So when I did my research in Australia on these polar dinosaurs, the critters I was looking at were, they weren't latest Cretaceous specimens. So, the most famous fossils come from the Maastrichtian of the Cretaceous period. And those are things like Triceratops and Tyrannosaurus rex. The specimens from Australia that I had the privilege to study were from the early Cretaceous, within the Aptian and Albian ages of the Cretaceous, so about 125 to 100 million years ago. So during this time, during the Aptian and Albian, the land masses of Australia and Antarctica were beginning to drift apart, and during this time, the state of Victoria, which is presently in southern Australia, the southernmost point of Australia on the eastern coast, Victoria was within the Antarctic Circle. So the Antarctic Circle is a region sort of like the North Pole. The Antarctic Circle is near the South Pole, and within the Antarctic Circle, you get months of darkness as well as months of sunlight. So during the Cretaceous period, there wasn't ice sheets, like permanent ice sheets on the North and South Poles as there are today. But that doesn't mean that the Antarctic circles weren't cold. They were, they had lower annual temperatures than, lower latitude areas. Imagine something like an Oregon or Washington type climate with the forests, very damp, cool areas. And likely in the wintertime there was some snow. Of course, it wasn't permanent snow, but it did get cold enough for snow. So with an environment like this, cool, sometimes it snowed, months of darkness, months of bright sunshine, the dinosaurs, as well as everything else that was living there, were living in pretty unique conditions compared to animals living at lower latitudes across the planet. So if these environments were so unique, were the dinosaurs that lived there also unique? What kind of adaptations did they have to enable them to succeed in these very interesting, different environments? So these were the questions that I had, since I like to study dinosaurs and how they grow. So I traveled to Australia, and I was very interested in taking a look at the fossils that were coming out of the Cretaceous sediments there to try to answer these questions. And I was lucky enough to get to go to the sites where some of these fossils have been excavated. And when I went to Australia, it was my summer, so the northern hemisphere's summer, the southern hemisphere's winter, and the cliff faces that these fossils come from are very dangerous. They are constantly being eroded. The tides are, you have to keep track of the tides in order to get to the fossils to excavate them. So the dinosaur fossils in Victoria, Australia are not really easily accessible, not something like, the scene from Jurassic Park where you're just taking a brush and brushing off dinosaur fossils. These took a lot of work to get to and a lot of effort. But the effort is really well worth it, because the dinosaur fossils are tough to get out of the ground, but they can be very amazing. You might know that Australia is very famous for its opal mines, a very pretty, precious gemstone. But one of the amazing things is that some of these mines actually, occasionally they'll find dinosaur fossils in these mines that have been replaced by opal. So you get basically dinosaur bones, opal in the shape of dinosaur bones, and they're just absolutely gorgeous. More relevant to what I'm working on are dinosaur fossils that are still articulated and encased in the rock. And so over years and years and years of collecting specifically the dinosaur fossils from the coastal localities, the Museum Victoria has amassed quite a decent collection of these fossils. And the fossils that I'm going to talk about, the dinosaurs that I'm going to focus my conversation on today, aren't really flashy or amazing. So a lot of people, when they give talks about dinosaurs, they're focusing on the really cool ones, the ones that people think are really cool. So Triceratops, T-Rex, the bone-headed Pachycephalosaurs, the long-necked sauropods, the vicious raptor dinosaurs, and even the duck-billed dinosaurs. Some people, including me, think that they're really cool, and they're really interesting. And unfortunately, the dinosaurs that I'm going to talk about today are frequently overlooked. They're not very famous, and chances are a lot of you probably haven't heard of them, at least a lot of you over a certain age. So these guys they even have an impossible name to pronounce, which makes them even more obscure. These are grouped as a term, they're called Hypsilophodontids. And this is what we call in paleontology a wastebasket term. Basically, these are small-bodied, basal, or you can think of as primitive ornithopod dinosaurs. So they're small, they don't have any flashy teeth, they don't have any flashy claws. They're these little bipedal, plant-eating animals that, haven't been that well publicized in the popular literature or anything as far as general scientific announcements or research on dinosaurs go, just because I think they're just not that interesting to look at. They don't have like really cool headgear or sharp teeth. They didn't hunt in packs, they ate plants. So they're largely overlooked. But the really cool thing is even though they're small, the smallness I think is really helpful. Imagine they're about the size of, like, if you're Australian, you would call it a wallaby. If you're from North America, you can imagine them being about the size of a large turkey. And they're really cool because of their sample size. So a lot of times you've actually probably seen these in exhibits and just not known about them, or maybe you overlook them yourselves because they're never the highlight of an exhibit. They usually end up as filler for exhibits, showing off the cooler theropods that are eating them. So they'll be the target of some kind of predatory attack. They end up in the mouths of theropods. So they are pretty abundant on every continent, and their sample size is decent pretty much everywhere. And that's really important for answering my questions specifically about the polar dinosaurs in Australia. They're tiny, so they were too small to have migrated out of the polar environment. So within that Antarctic Circle, where you get months of darkness, months of light, cooler weather, occasional snow, they wouldn't have been able to make really long migrations to avoid this kind of annual change, this harsh winter, and harsh in that you're experiencing dark or dusk for months on end. And then I keep going back to the sample size. These little basal ornithopods or Hypsilophadonids are pretty common in the excavations for the Australian fossils of Victoria. And what I'm really interested in are these large sample sizes, because if you think about a population, there's a lot of variability. You think about humans are a great example of that. We don't all look alike. We are taller, shorter, thinner, more robust skeletally. So there's a lot of variation within a population, and the more you sample from that population, the more you get an idea of that variability. And this is really important when you're trying to reconstruct how an animal as a species was actually growing, because if you accidentally, without knowing it, sample one individual that happened to be growing really slowly or really quickly, you could interpret that for the entire species, and you might be incorrect in your interpretations. Now, for these dinosaurs, these Hypsilophadonids, they have been studied before. So the ones from Victoria. Back in the late 90s, a study was done looking at the bone microstructure of these little Hypsilophadonids. And back then, a single bone of the Hypsilophadonids was used to try to infer the growth history of these little guys. And back then, you have to realize that histology was still fairly new. The idea of taking a bone and slicing it open to look within it was a bit terrifying back then. It's become very commonplace today, but back in the late 90s, it was still sort of a new idea, sort of gaining ground in paleontology as a research tool. So the study examined the bone tissue of this little dinosaur, this Hypsilophadonid, and they saw that the tissue didn't have any interruptions in it. And interruptions in tissue look like tree rings. So if you imagine chopping a tree down, you see concentric rings in the tree trunk. Well, if you cut a bone, a long bone, a bone shaft, transversely or across, you'll see the same thing microscopically if the animal stops growing every year. So just like trees, the tree rings are formed because the tree stops growing every year. Well, animals today do this as well. So the study in the 90s didn't find these rings in the little Hypsilophadon bone. So the authors concluded that this must mean that these little guys did not hibernate during the polar winters, that they just kept on living and eating and growing, and that the polar winters didn't affect them in any particular way. So that was the hypothesis that was proposed. And after reading this paper, in the 2000s, I wasn't really sure if this was the correct interpretation, because as a paleohistologist, I've been looking at a whole bunch of different dinosaur fossils as well as modern bone. And if an animal took more than a year to get to adult size, so think about humans, we reach adult size in our late teens, early 20s. Animals that take multiple years to get to their adult size stop growing annually and produce these rings. We're still not really sure why, because it doesn't really seem to have anything to do with, annual stresses, because we can, again, humans, we don't really have those annual stresses, like, we're not typically exposed to extreme conditions. We're pretty much living comfortably. We have ample food, we have, shelter, but we still produce these growth rings annually. The cool thing about these growth rings is that because they are annual, you can count them and figure out how old an animal was when it died. Well, I see these I see these rings, these growth rings, in all kinds of different bones, from fossil extinct animals and animals that are alive today. And the fact that this study from the 90s didn't find these growth rings in these dinosaurs was a little surprising to me, because I don't know, or I didn't think that it had anything to do with hibernation, with the behavior, because I see it in animals across the planet. So, we've got rings in polar bear bones, for example. We've got rings in elk bones, for example. Animals that we know don't hibernate have these rings. So I wasn't sure exactly how these rings would tie into hibernation in the first place. So this is a great example of science in action and hypothesis testing, because scientists will propose a hypothesis. In this case, I expect to find growth rings in this dinosaur bone. They did not find growth rings, therefore they proposed a hypothesis that it must be because these animals weren't hibernating. I'm interested in testing that hypothesis because I'm finding the opposite. I'm finding growth rings in all different kinds of animals, both those that hibernate and those that do not. So I don't think that these growth rings really have anything to do with hibernation. So that's my hypothesis. And that's how science keeps moving forward. You propose a new hypothesis, you test it, and then you see what the results are. So that's sort of where I was coming from, having read this literature. And I was lucky enough to receive a National Science Foundation grant to travel to Australia. And I got in touch with Tom and Patricia Rich, who work at the Museum Victoria, the Melbourne Museum specifically, and are the ones that have been leading these excavations to dig up these Cretaceous Hypsilophadonids on the coasts of Victoria for many, many years. And they were on board with me coming down to Australia and actually studying more of their fossil specimens. And this was great news to me because, previously only a single specimen had been studied in the way that I study it with histology. So I could then add to this sample size, increase it, and see exactly what was going on. And so this was super exciting. I, went down there. I learned all I could about these little Australian polar dinosaurs. And what I'm particularly interested in is sampling the same bone from multiple individuals. So with paleohistology, we want to get consistency so that we can sort of compare across different individuals. And the best bones for looking within at the bone tissue microstructure are the hind limb bones, because they take most of the weight of the animal. So as the animal is getting bigger, as it's adding weight, these bones are changing to compensate for that. Therefore, think of it as these bones being the record keepers of the animal's growth. So a hind limb bone, or hind limbs, consist of the thigh bone or the femur. Then below that, below the knee, you have two bones, the tibia and the fibula. And then below that, you've got your ankle bones and your foot bones. Well, for me, I'm really interested in studying the histology, or the bone tissue microstructure, in the tibia. So I was really lucky to sample all these little Hypsilophadonids, because it's still sort of hard to get permission, because again, you have to cut through the bone. But because histology and what it can tell you has really started to gain traction, it's really been a bit easier to do this. And so I was given permission to histologically sample tibiae from the collection at the Melbourne Museum. And I was able to sample 16 individuals total. So that brings the sample size up from 1 to 16, which is just super amazing. That might not seem like a lot, especially to any biologists or ecologists out there, but for dinosaur paleontology, anything above a sample of 1 is oftentimes just amazing and totally exciting. So in order to do this, in order to histologically sample the specimens, I would like to take a few minutes and just walk you through the process that I use, because again, I am cutting into the bone, which might sound terrifying to some of you, and I want to try to explain why this is worth doing. So with these specimens from Australia, first off, I remove a piece from the bone shaft, and I want to make sure that the shape of the bone, the overall shape of the bone that I have, that I'm going to alter, is preserved as well as I can. So with the piece that I've removed from the shaft, I make a mold and a cast of it. And to make a mold, it's usually a 2-part mold. So you will cover part or 1/2 of the piece that you've taken out by some sort of putty or silicone gel, and that eventually takes the shape of half of the bone. Then you flip it around and you do the other side to make a lid or a cap for it. And I did this for every bone in my sample in Australia. And what you end up with is a void where that original sample was. Then you can pour in some sort of casting medium. So a lot of you might have done this with plaster of Paris before. You can use that. You can use plastic resins, something that will fill in that void and basically produce a replica of the piece that we took out. That way we have a piece that looks just like the original, the replica, that you can glue back in. And the really cool thing is that some museums, like the Museum Victoria, wanted me to actually paint those cast replica pieces and reinsert them into the bone. So the way I do this is to just use some acrylic paints, and once I've matched the color of the fossil, I can take the painted replicas and glue them back into the bone, and it's sort of like nothing ever happened to them. The same shape is preserved, the length is preserved, and in order to indicate that these pieces are replicas, so someone in the future might want to do some histology on these specimens, in order to make sure that they don't cut into plastic or resin, I like to paint little yellow dots on one side of the bone just to indicate that these are the replica painted pieces. And I remember really feeling proud about myself because I had sampled all 16 bones. I had made the molds and casts. I had reinserted the pieces, the replicas, and Dr. Tom Rich was coming by my workstation at one point, a couple of weeks into the research project, and asked me if I was ever going to get to it, if I was ever going to get started, cutting these bones. And what he didn't realize is that I was already finished with taking those pieces out, and he was looking at the replica pieces. So that made me feel really good that, it passed a curator's inspection. So the whole point of that is just to say that, yes, we are removing a piece, but we can make molds and casts to replicate the shape. We can even nowadays use CT scanning or laser scanning to get pretty much accurate 3D printable pieces from the bits of specimen that we've removed. So now that I've got my piece out, I need to prepare it for histological analysis. And this is a really long, involved process because, again, we are taking out a piece that you can't replace. It's a piece of fossilized bone, and we want to make sure that we get all the information we can out of it in as carefully as possible. So after getting those pieces of bone out or separated from the bone shaft, the next step is to embed them in a plastic resin. So we've got this polyester resin we use, and we put the bone in a container. The polyester resin starts as a liquid, which then, once you add a catalyzer, it will harden up over a period of 24 hours. And so basically what you end up with is a little piece of bone in hardened plastic resin. It kind of looks like one of those paperweights that you always see with like the little insect or something in it, except this time it's a bone. And we want to do this because in order to make our thin section slides to eventually look at the bone tissue, we have to take thin wafers out of the bone. And if we were just to run one of these bones through a tile saw, it would just crumble because, again, it is fragile bone. It's a fragile fossil. So the resin helps stabilize and harden the bone because the next step is to take a wafer out of the bone. And there's several ways we can do this with various size saws depending on the specimen size. So a typical wet tile saw is my go-to power tool, and I'm probably the only lab on campus, I'm pretty sure I'm the only lab on my medical campus that actually has 3 different types of saws in the lab for cutting fossils. But we use a continuous rim diamond blade, or we can use a bandsaw for the much bigger specimens with a continuous a diamond blade. And what this does is cut these nice wafers for us that are maybe, 3 millimeters in thickness, maybe a little thicker. But what we end up with are basically little slices of the bone shaft, which we can then glue to a slide. And by a slide, that's just, a piece of plastic or glass. You glue the specimen to it, and then that's when the work really begins. So you've got these pieces of bone, they're glued to these slides, and then you just start polishing. And we use a lapidary wheel for this, and we start with really coarse grit paper. So silicon carbide paper, we usually start with 60 grit, which is really rough, and as the bone slice is getting thinner and thinner, we go with finer and finer grit paper. Basically, we start with the 60 grit, we have lots of deep scratches, and we remove those scratches by going finer and finer grit until the specimen is usually between 60 and 100 microns in thickness. So, in some cases less thickness than a human hair. And at that point, we have bone thin section slides that are thin enough for light to pass through and for us to take a look at under the microscope. And what's really cool about this process is that for me, histology is really exciting because you have this fossil bone that you can hold and look at, and it's millions and millions of years old, but then you can take a look at the microstructure inside that bone, and it's preserved just as well as the outside. So everything that was contained inside the bone is just as well preserved as the bone itself. Super exciting for me. And so this is what I do as a paleohistologist, is look at the bone tissue of oftentimes these huge animals, but I use a microscope to do it, and I learn about their growth that way. And so when I do this, I take a bone thin section, and for fossil bone, it's usually been stained through just the minerals percolating through it or perforating through it. And so you'll have basically a marrow cavity in the middle, which is a circle, and then you'll have sort of just this ring of actual bone, from the marrow cavity out. And when you are looking at it under a microscope, the light's going to pass through the marrow cavity, making it pretty bright, and then all the bone is going to look a bit darker. And when I'm looking at a bone thin section or a slice, I'm looking basically at a cut or transverse cut through a bone. So, if my hand here is a bone, I've cut it this way so that I'm looking at the bone on end. So the marrow cavity is in the middle and all the bone is encircling it. So if you ever had like a ham steak from a thigh, you always have that round little bone in the middle. That's the same concept. That's what I'm looking at. Now what's amazing to me is that we can use modern animals and take a look at their bone tissue. We have to understand what the extinct or the fossilized bone tissue is telling us. In order to understand what bone says in general, you have to have an animal that you observe today, you have to have an animal that you understand the biology pretty well, and you have to have an animal that you know its life history. So what year was it born? What year did it die? Did it get sick? Was it healthy? Was it a male or a female? So if you know this information about an animal today, you can look at its bones and try to correlate or connect what you see as far as the tissue patterns with what that animal was going through when it was living. And so when, for example, if you take a look at an elk bone, you can see the blood vessel canals. So where the blood vessels were invading the bone, you can see that. You can see the specks where the bone cells were growing and producing bone. The really cool thing that a lot of people don't think about much is that bone is actually a living tissue. Normally you think about bone as, just something to hang muscle on. It provides the strength and the structure for the shape of the animal, but it is a living tissue. You have these bone cells. Their whole job is just to secrete bone, and then they get stuck or buried in the very bone that they produce. But they're still alive, they're still in there, so blood vessels have to bring these bone cells that are just stuck there. The blood vessels are bringing them nutrients. And the more bone cells you have in a tissue, the more blood vessels you need. The more bone cells you have in a tissue, the faster bone can be produced. So just by looking at the amount of blood vessels and the amount of bone cells under a microscope, you can already start deducing relative growth rates. Was this animal growing fast? Was it growing slow? Did it start out fast, and then as it was getting older, did it start growing more slowly? And of course, you can also see those really cool growth rings. So, for example, an elk bone has fast-growing tissue. It's got lots of blood vessels, lots of bone cells, and it also has growth rings. Every year, elk stop growing for a couple of months. And this is just a natural thing that all vertebrates seem to do if they take more than a year to grow up. Now, the really cool thing is, if you understand this and you can read the bone record of modern animals, taking a look at fossil bone tissue looks exactly the same. And I know it sort of sounds silly, it's just kind of mind-blowing to me. Bone is bone. So the bone in me has the same composition and structure as the bone in an elk, as the bone in an alligator, as the bone in a dinosaur. It's just that the bone cell arrangement, the blood vessel arrangement, that's the difference. But the actual bone composition is the same, which is just so cool. So a dinosaur bone, for example, one of these small-bodied Hypsilophadonids from Australia, has blood vessel canals. It has bone cells, or we find the pits where the bone cells used to be, because most of the time, the soft tissue has degraded away. But evidence of those bone cells are there because they made themselves little homes. They secreted all the bone around them, and then they had their little homes to live in. So we find the holes where the blood vessels used to be. We find the little pits where the bone cells used to live, and we find growth rings. And again, because we know growth rings are annual in animals today, they form on an annual basis, we can count those in an extinct animal and get a good idea of how old it was when it died. And this is just so cool to me. And again, as I was saying with the elk, you can determine relative growth rate. Was this animal growing slower or faster than another animal? And today we tend to find really richly vascularized bone tissue with lots and lots of bone cells in warm-blooded, fast-growing animals like an elk or like a big bird like an ostrich or a rhea. On the other hand, we find prevalence of slowly deposited tissue in something like a reptile, like an alligator. Alligators do take many, many, many years to reach their huge adult sizes, but they typically do it very slowly. And so when bone tissue is deposited slowly, you don't get as many bone cells and you don't get as many blood vessels, because you don't need that much nourishment if you don't have as much cells. And the bone cells are not rapidly producing bone tissue. It's being produced at a more orderly rate. So typically, slowly deposited bone also looks really layered. It looks very organized. Now, of course, alligators take many years to reach adult size. They have growth rings, just like elk. So growth rings don't really have anything to do with metabolism, whether or not you're warm-blooded or cold-blooded. Again, it just seems to be something that is inherited from a common ancestor of all vertebrates. And so we see these across the board in a bunch of different animals, both living and extinct. Now, when I was looking at the bone tissue of these polar dinosaurs, I got to see all these things. I got to see, bone tissue that looked like it was fast growing, bone tissue that looked like it was slow growing, growth rings. And I wanted to sort of walk you through the process so that you could understand what it was that I was trying to look at and see under the microscope, and also so that you will hopefully understand by the end why histology is so important to use in paleontology, and why we can learn so much more by sampling these bones. And so I thought, after telling you about the procedure, that I would tell you about this really cool story that I learned just by looking at the bone tissue of one particular Hypsilophadonid from Australia. Now, this guy, Junior, is what they nicknamed him, or it. It's basically the back end of one of these Hypsilophadonids from one of the sites on the coast, and it consists of a connected or articulated couple of legs. So the left and the right leg of this dinosaur were preserved in place with the thigh bone connected to the tibia bone. And when I look at bone tissue, particularly the tibia in this case, what I normally see is sort of a loosely triangular shaped bone with a nice circular marrow cavity. The bone is this nice honey brown, and I can see the bone structures inside, but it looks like a typical columnar bone. Well, Junior, one of its legs was injured. One of the tibiae, or the shin bones, on one side, one of the two, was injured in life, and so it developed a pathology, or an unusual bone growth. This doesn't look like a normal tibia, and so there's been some studies on this particular dinosaur, Junior, and it turns out that this one particular tibia actually was shorter than the other. So this poor guy would have actually had a limp in life. And so what I wanted to do is take a look at the histology of this pathologic tibia and try to learn a little bit more about the life of the tough life of Junior. So I was permitted to take a couple of samples from its femur, which looked perfectly fine. It looked like a typical femur should, and that pathologic tibia. And when I took a look at the pathologic tibia, remember, a normal tibia should just look sort of triangular in shape, very smooth edges. The tibia of Junior was an absolute mess. I have an image of this tibia, and I've outlined the normal looking bone, I guess you would call it, in blue, just so it becomes more visible. But everything else that's not outlined in blue is abnormal. It shouldn't be there. So what this is telling me is, first of all, I see two halves of this tibia bone, which means that it was likely fractured in life. It was just sort of sheared across. And this injury then produced probably infection. And so this made the bone just go crazy as far as growing. It wasn't growing to any particular pattern. It was just sort of growing all over the place. And so this would not be a natural shape for a tibia. It was probably very debilitating for this animal. It was probably very painful for this animal. So what I could see from the normal tibia tissue in the middle of this bone were 4 of those growth rings. So what this tells me is that this animal was at least 4 years old before it did whatever it did to hurt itself and cause this pathologic growth. So then I took a look at the femur, and the femur should look, again, like a round column with a marrow cavity in the middle, but as is often the case with fossils, through just the process of burial and compaction, part of this bone was crushed in on itself. That's unfortunately more often than not what we find when we look at histology, but there was a nice half of the bone that was still nicely preserved. It had some cracks running through it, but I could see enough of the tissue to get more of the story. So when I looked at the undeformed portion of this femur, what I saw were 2 growth rings that were normal looking, as in they were nicely spaced apart, but then 2 more growth rings, which I highlight in blue, were very closely spaced together. And the spacing of growth rings tells you how much growing the animal did from one year to another, right? Every year the animal grows, then the animal pauses its growth, causing a ring, then it resumes growth, and so you get this cyclical pattern. But with the two growth rings that are really close together, that shows me that growth stopped, then there wasn't much growing during the preceding year, and then growth stopped again. So this could be an indication of some sort of sickness in that the animal wasn't getting enough nourishment to grow. Maybe its body was diverting some resources that would normally be put into growing to fighting off infection. It just looked very unusual because this sort of close spacing of growth rings in the middle of the bone isn't something I'd seen with the other samples. And then after that, growth seemed to sort of resume normally again with some more widely spaced growth rings closer to the bone surface. So my hypothesis is that these two closely spaced rings suggest maybe this is the point in which, in this dinosaur's life, that this injury and sickness occurred. And again, interestingly enough, these two are the fourth and fifth growth rings. So remember the pathologic tibia had 4 growth rings in it before growth started going crazy. It's interesting that I find that this spacing, this closely spaced year of growth happened between year 4 and 5. So I think that might be some evidence to support this idea. But regardless, what this tells me is that this animal got injured, it got sick, its bone got infected, but it continued to live for 3 years after the fact. So it was going around with this infection, had to be in so much pain, but it lived for 3 years. And so that is just a really cool story to be able to tell. I mean, it's really sad that this poor little guy ended up, dying, but and probably in a lot of pain. But this is a really interesting story that we can tell just by looking inside the bone. We knew that it had a pathology. We knew that just by looking at the bone in hand sample that something was wrong with it, but we had no idea before actually looking at the histology how old it was when it might have gotten injured, and that it actually lived for several more years after the fact. So that just was so cool to me that we could learn a little bit about this poor little guy's life 120 million years ago. So that was one really cool thing that I could do with the research from Australia. But what I was really there to do was to produce a growth series, a histological growth series. So to find the smallest individuals all the way up to the largest individuals, to try to get the whole story of how this particular group of animals was growing. That's why growth series is so important. You get individual variation as well as how the animal is growing from year to year. So the smallest bone that I was permitted to sample from this site was it was really adorable. It's so tiny. It had to have been from a hatchling. The ends of the bones were underdeveloped, which means they were cartilaginous. It had this tiny little process on it. So the process on the femur attaches to muscles from the tail. And it was just this tiny, cute little nubbin of a thing. And when I looked at its histology, I was able to see that this tiny little dinosaur had this amazingly vascularized bone. And the bone tissue organization was very disorganized. It was ropey looking. It was almost what we call woven. And when we see this type of tissue, we know that the animal that was producing it was growing very quickly. Again, based on animals and their bone that we look at today. So I was able to look at that and learn that when these tiny little Hypsilophadonids hatched out, they basically hit the ground running in terms of bone growth. They were growing very quickly, very early on. And one other really cool thing that I was able to do was resample the single bone that was sampled back in the late 90s from which that first study was based on. So it was a femur, and you can see that the process that attaches the femur muscle to the tail is way more developed here. It's definitely an older individual. And when I histologically sectioned it, it was a very nice, beautiful triangular shape with a very large marrow cavity. And what really excited me was the fact that I found growth rings. So in the specimen that was reported in the late 90s not to have these growth rings, they're actually there. They were just overlooked. And what's really cool is that there were several of them. So this animal lived and was growing for many years before reaching adult size. And this by itself then falsifies that hypothesis that these tiny little Hypsilophadonids hibernated or didn't hibernate to get through the winter. This falsifies the idea that growth rings have anything to do with hibernating or not hibernating. So this is basically science in action. I tested an earlier hypothesis to see if it held up. I replicated the study and I found growth rings which weren't found before. So that was super exciting to me. And by using a growth series and taking a look at these cross sections of bone, I was able to reconstruct how these little Hypsilophadonids were growing and even how their bone was changing shape over many years. So in an older bone, the marrow cavity is expanding, so you lose a lot of that early record of growth. And you can still trace those earlier rings, it's just that they are oftentimes destroyed by that marrow cavity expansion. So if I outline the marrow cavity in pink just to keep an eye on it, then I can trace every growth ring in the bone in blue, for instance. And by doing this, I can get this visual idea of how annually the bone not only was changing shape, but how much growing was occurring at different parts of the bone every year. So some areas of the bone had a lot of growth, other areas weren't growing as much from year to year. And this is how you change the shape of a bone through growth, which is just super cool and exciting to me. Another hypothesis I had the chance to test was whether or not the Hypsilophadonids were all juvenile. So one hypothesis that was put forward in the early 2000s is that in a lot of cases, these Hypsilophadonids were just juvenile animals, and they might have grown into something the size of an iguanodon. This is my kitty. He says hello. Every time I try to do anything as far as recording, he has to make an appearance. So anyway, the hypothesis was that maybe these Hypsilophadonids were juveniles of something like an iguanodon, something much bigger. And in order to determine whether or not an animal is an adult, you can look for a particular type of bone at the bone surface. And again, we know this by looking at animal bone today. So if an animal is skeletally mature, if it's reached its adult size, it forms something called an external fundamental system. We like to call it EFS just because it's a lot easier. But this EFS tissue is on the bone surface, and it looks completely different than the growing bone underneath. And it basically signifies that there's no more growing in length. It's just that the bone tissue is sitting there just sort of thickening up. And so we find this, for example, in alligators, showing that they actually do stop growing. We also find this in mammals and in birds, such as an ostrich. So I was on the lookout for this EFS structure as well. And I found it. So the EFS in many cases in these little Hypsilophadonids were actually really well developed, meaning that these particular animals, some of them had been at their adult size for a very long time before they died. And so this shows that, or falsifies the hypothesis that Hypsilophadonids were in large part juveniles of something else. Hypsilophadonids, these little basal ornithopods, actually were, small-bodied animals that didn't get much bigger than a wallaby or a turkey. So again, really cool. And so with all of my sample, I was able to construct somewhat of a growth curve by counting up the growth rings in each individual I looked at and plotting that against the length of their tibia. So, for example, a tibia might have 3 growth rings in it and be 30 centimeters long. So I put a dot on the graph that way. And so in general, using 16 samples, I can conclude that these little polar Hypsilophadonids were growing the fastest during the first 3 years, and then they sort of started slowing down, which is what most animals do. They have an early period of fast growth, then growth starts slowing down, and then they reach their adult size in about 5 to 7 years. So they got to be about wallaby or turkey size in about 5 to 7 years. And so this is information that we had never known before about these little polar dinosaurs from Australia. But what I really want to point out here is that the sample that I was given access to was a combination of 16 individuals from both the Aptian and the Albian time spans within their Cretaceous. Now, if you remember, that's about a 12 million year time gap that we're looking at here. And because of that, looking at that graph, it's probably likely that I'm not looking at the same species. That samples from one tibia and samples from another tibia are probably different species. But what's really interesting is that even though the species may have changed in those millions of years, the growth trajectory was pretty similar, which means that even though we might be evolving different species of Hypsilophadonids through those 12 million years of time, that growth trajectory was very successful for surviving and thriving in these polar conditions. So even though the species might change, the way they were growing was pretty much staying the same. And so my next question then was, all right, now that I've got this information about these dinosaurs and how they were living in Australia, how does that compare with their relatives living in other places around the world? Well, I took a look at Hypsilophadonids because, again, like I said, they're very diverse. They're found everywhere, in a whole bunch of different regions, not just North America. We find them in Europe and Asia and so on. So I looked at some the literature for Hypsilophadonids. I looked at some thin sections specifically from Montana, and the really cool thing, there's pretty much no difference between the growth trajectories reported for wallaby-sized Hypsilophadonids in Australia and wallaby-sized Hypsilophadonids elsewhere in the world. And, at first when I came across this, you can compare the histology of a Hypsilophadonid from Australia to that from Orodromaeus from Montana. They look pretty much the same, exactly the same amount of vascularity, the rapid growth early in life, same small body size. It at first seemed kind of disappointing to me because I really thought, oh, this is a really cool, unique environment. These polar dinosaurs should be doing something really cool. They should have, evolved a really cool strategy for coping with this kind of lifestyle or environment. But there's no difference. And then I started thinking about it, and actually, that in of itself is really awesome. That means that, during the Jurassic, when we had these little Hypsilophadonid precursor things running around, dispersing to the different continents, they already were basically, their physiology was pre-adapted to be successful anywhere in the Mesozoic world. And thinking about that is just amazing, because that shows you how successful dinosaurs actually were. Of course, they might have evolved, they probably evolved different behaviors for coping with the polar winters. They may have had a different kind of insulation, maybe thicker insulation for living at the poles. But as far as the histology goes, their growth rates and their metabolic engines were exactly the same from those in lower latitudes, which is just super cool. And I did want to say that, growth rings do not equal hibernation, and my research demonstrated that. But that's not to say that these dinosaurs living at the poles didn't hibernate or do something behaviorally to cope with the winters. It's just that histology can't show us that. Growth rings are not an indicator of hibernation, because they are pretty much universal across latitudes, and don't relate to that specifically. But that's not to say that these dinosaurs didn't have some sort of adaptation for coping with this unique environment. And then lastly, I did want to say that I was able to take a look at a couple of theropod bones from the Melbourne Museum from polar latitudes. And because the theropods are normally rare in a bone bed, we didn't have as many to sample. I didn't have as many to sample as far as just being accessible, but I did get to take a look at some Teminous bones. And Teminous was a small theropod living the same time as these Hypsilophadonids. It was originally thought to be an ornithomimid, but more recently it's turned out that it's probably a tyrannosauroid, which in my book makes it even more cool. But when I took a look at its histology, again, just like with the Hypsilophadonids, the histology of Teminous didn't look much different from that of lower latitude small-bodied theropods. So I compared it, for example, to a Troodon, which isn't really closely related to Tyrannosaurians in general, but the way the Troodon was growing was very similar to the way Teminous was growing, so their histology was very similar. And so this again shows me that, whatever the ancestral theropod had as far as its physiology and adaptations go, was perfectly suited or pre-adapted Teminous for living in these cooler polar environments, which again I think is just super amazing. So with that, I just wanted to leave you guys with the idea that hopefully you've learned a little bit more about polar dinosaurs, seen how amazing histology can be and how important it is, and that it's totally worth, I think, the thin sectioning process to learn something by looking inside the bone. But I also want to, or I hope that you have also learned something about the scientific process. Maybe there are histological signs of behavior that I haven't been able to recognize because there's still so much to do and learn from modern bone histology. And that this is an example of how science works. You propose a hypothesis, you test it, then you either reject it or fail to reject it, and then you repeat the process. And it might sound silly, but I'm really hoping that maybe one day one of you will use histology to test my hypotheses and maybe even show me something new, because that's how science works and that's how science progresses. And I think that there's still so much more to be done, especially with polar dinosaurs, because they were living in such a unique environment. And I would encourage you guys to look deeper into this and again, always ask questions. Learn as much as you can, and you'll never be disappointed with the answer.